Welcome, everyone. Today, we are diving into one of the most elegant and vital processes in all of biology, oxidative phosphorylation. If you have ever wondered why we need to breathe oxygen every single minute of our lives, the answer lies right here. This is the grand finale of cellular respiration, the powerhouse mechanism that keeps our brains thinking and our hearts beating. Let us begin our journey into the microscopic world of the mitochondria. To truly understand how this works, imagine a massive hydroelectric dam. A dam holds back millions of gallons of water in a high reservoir. When that water rushes down, it spins giant mechanical turbines to generate electricity for entire cities. In your body, oxidative phosphorylation works exactly like this microscopic dam. Instead of water, your cells pump tiny charged particles called protons across a membrane. When these protons rush back through a special protein turbine called ATP synthase, they generate ATP, which is the biological electricity that powers your entire body. Now that we have our dam analogy in mind, let us look at where this actually happens. Oxidative phosphorylation takes place in the inner membrane of the mitochondria, famously known as the powerhouse of the cell. It is the final stage of cellular respiration, taking the energy extracted from the food we eat and converting it into a usable form. The entire process is built on two deeply connected components working together: the electron transport chain, which acts as the pump, and chemiosmosis, which acts as the turbine. Let us zoom in on the first of those two components, the electron transport chain. High-energy electrons are delivered by carrier molecules like NADH and FADH2. These electrons are passed down a series of protein complexes, almost like a microscopic game of hot potato, until they finally land on oxygen to form water. As these electrons fall to lower energy states, they release energy. The cell brilliantly uses this released energy to pump protons out of the mitochondrial matrix, building up that high-pressure reservoir we talked about earlier. So we have pumped all these protons out, but what happens next? This brings us to the second component, chemiosmosis. The inner mitochondrial membrane is highly impermeable, meaning those pumped protons cannot just leak back across. They are trapped. Their only way back into the matrix is through a highly specialized protein channel called ATP synthase. As the protons rush through this channel, their movement physically causes the base of the enzyme to rotate. This incredible mechanical rotation provides the exact energy needed to jam a phosphate group onto ADP, synthesizing the ATP that your cells crave. Understanding this mechanism brings us to a crucial question. Why does this complex process matter so much to our survival? The answer is efficiency. Oxidative phosphorylation is responsible for producing roughly 90% of the ATP your body uses. If we only relied on earlier metabolic steps like glycolysis, a single molecule of glucose would give us a measly 2 ATP molecules. Thanks to this mitochondrial dam, that same glucose yields about 30 to 32 ATP. This is why poisons like cyanide are so deadly. They block this exact pathway, instantly shutting down the cellular power grid. To truly appreciate this power grid, we must look at the mathematics behind it. How much energy is actually available to pump those protons? We calculate the energy released by the falling electrons using the Gibbs free energy equation for redox reactions. In this formula, delta G prime naught is the usable energy released. The variable n is the number of electrons transferred. F is Faraday's constant, representing the electric charge of a mole of electrons. Finally, delta E prime naught is the reduction potential difference, which measures how strongly the final acceptor pulls the electrons. When this voltage difference increases, the free energy becomes more negative, meaning a much larger amount of energy is released to pump protons. Now that we have our formula, let us see this with a concrete example. During oxidative phosphorylation, the molecule NADH donates 2 electrons to oxygen. We are given a standard reduction potential difference of 1.14 volts, and we need to calculate the standard free energy change in kilojoules per mole. We choose the Gibbs free energy formula because it directly connects electrical potential to chemical energy. We plug in 2 for the number of electrons, 96.485 for Faraday's constant, and 1.14 for the voltage. Multiplying these together gives us negative 219.98 kilojoules per mole. The negative sign confirms energy is being released. Since making 1 ATP only takes about 30 kilojoules, this single NADH provides enough energy to synthesize several ATP molecules. This massive release of energy we just calculated is used to pump protons, which brings us to our next major concept. Once the protons are pumped across the membrane, they create a biological battery. The total pressure pushing the protons back through the turbine is called the proton motive force, denoted by delta P. This formula beautifully shows that the driving energy comes from 2 distinct sources. The first term, delta psi, represents the electrical charge difference, because protons are positively charged. The second term involves delta pH, representing the chemical concentration difference. If either the electrical charge difference or the acidity difference increases, the total driving force increases, allowing the cell to manufacture even more ATP. Before we visualize the whole system, let us calculate the exact pressure of this biological battery. A researcher measures a mitochondrial membrane and finds an electrical potential of 0.15 volts. The outside is more acidic, giving a delta pH of negative 1.4. We are asked to find the total proton motive force, assuming the temperature constant term evaluates to 0.059 volts. We use the proton motive force equation to combine the electrical and chemical gradients. We plug in 0.15 for the charge difference. Because the outside is more acidic, the negative delta pH cancels the negative sign in our formula, which makes physical sense since both forces are pushing protons inward. Adding the chemical portion of 0.0826 to the electrical portion gives a final answer of 0.2326 volts. This combined voltage is the exact pressure forcing protons through the turbine. Now that we've calculated the forces at play, let us look at the complete picture. In this diagram, you are seeing the inner mitochondrial membrane. On the left is the electron transport chain. Notice the protein complexes labeled 1 through 4 embedded in the membrane. You can trace the red path of electrons, starting from NADH, flowing through the complexes, and finally landing on oxygen at the bottom to create water. As those electrons flow, look at the blue arrows pointing upward. They represent protons being pumped into the intermembrane space, building up that high concentration of positive charge at the top. Finally, on the far right, you see the green ATP synthase turbine. The protons flow back down through it, driven by the proton motive force we just calculated, physically spinning the enzyme to churn out ATP at the bottom. This is the biological dam in full operation. We have covered a lot of ground today, so let us summarize the most important ideas. First, Oxidative phosphorylation brilliantly couples the downhill flow of electrons with the uphill pumping of protons. Second, this pumping creates a biological battery across the membrane, measured mathematically as the proton motive force. Third, this combined electrical and chemical pressure drives the ATP synthase turbine to manufacture the vast majority of the energy your body needs to survive. You now understand the grand finale of cellular energy production and exactly how the food you eat and the oxygen you breathe are transformed into the energy of life. Great job following along with these complex concepts.